Good morning, everyone. Thank you for being here in person and also virtually. Today we have two of our T32 supported postdocs who are giving talks today. So there'll be two 20-minute talks, each one followed by questions. So our first speaker is Megan Ludwig. Megan received her undergraduate degree at Hope College in Holland, Michigan. And she was a T32 supported trainee there and also held a NIH NRSA. She then completed her PhD studies. She completed her PhD studies at the University of Michigan at Ann Arbor, where she was a T32 trainee and had an NRSA. Sorry, it's early for me, I guess. I'm just recovering from all the illnesses I had over the break. And she's been in Justin Drake's lab since about 2019, and is studying the role of exosomes in castrate-resistant prostate cancer. Our second speaker will be Julia Austin. Julia is a postdoc in my lab for about a year and a half. She received her undergraduate degree at the University of Arkansas at Fayetteville, and then finished her PhD at the University of Illinois in Chicago with Joanna Burdett, where she discovered novel plant steroid hormones that are ligands for the progesterone receptor. She was also a T32 supported trainee during her PhD work, and has been in my lab interested in the interaction between the glucocorticoid receptor and other family members, namely the mineralocorticoid receptor, sort of a new really fun project that we've gotten into. So without further delay, Megan will present first. Thanks, guys.
Thank you, Carol, for that introduction, and thank you to the Masonic Cancer Center series for letting me talk today about my project on looking at the proteome of extracellular vesicles. Let's see. There we go. Okay, so as Carol mentioned, I'm in Justin Drake's lab, where we focus on prostate cancer. I think the cancer statistics for 2023 will be out probably the next couple of days, but for now I have last year's statistics, which tell us that prostate cancer was estimated to be the primary cancer diagnosis for men in the US last year, and also the second leading cause of death. And so unfortunately, basically in short, that means that prostate cancer is an incredibly common and still lethal disease. And so in our lab, we focus on the lethal variants of prostate cancer, and these are resistant towards androgen and deprivation therapy. So this graph is a depiction of clinical progression over time. And so here we're representing PSA on the y-axis as indicative of androgen receptor-driven prostate cancer. Right? So a patient can present with a rising level of PSA, have a local advanced tumor, and then receive therapy such as surgery and radiation, and respond. However, resistance can develop, and so when it does, there are second-generation AR inhibitors, which can be effective, but again, frequently resistance continues to develop, and we end up with metastatic castration-resistant prostate cancer. And so there's generally two overall categories for this. One does correlate with rising levels of PSA, and so these cancers are still driven by active AR signaling, potentially through AR variants or other mechanisms. We also have aggressive variant prostate cancer, and so these are androgen receptor independent cancers. And so there's neuroendocrine and then also double negative. They don't have AR, they don't have the neuroendocrine markers, but we don't have any other positive way to identify them other by what they don't have. Okay. And so unfortunately, the aggressive variant prostate cancers are a growing issue for patients. The second generation AR inhibitors were approved for use in 2012. And so before 2012, most patients with metastatic castration-resistant prostate cancer had androgen receptor-driven signaling. And so that's this large green section on the pie chart. Post 2012, we're seeing much more of a blue segment, which is AR independent and neuroendocrine negative, and then also neuroendocrine positive here in the yellow. So we have a lot more mechanisms of resistance happening, especially in AR independent ways. And so the ways that I've chosen to study these mechanisms of resistance is through looking at extracellular vesicles. So extracellular vesicles, these are nanoparticles that are released by all cells. They could be found in all body type fluids, such as blood, urine, and saliva. And they are a lipid bilayer membrane, and they contain DNA, RNA, and protein that are released to communicate with other cells. These EVs can be exchanged through multiple different cell types. So this graphic is we have cancer cells here in the middle, and then other different cell types that can be exchanging EVs. So for example, we have migration and invasion EV promoting signals coming from cancer-associated fibroblasts. Mesenchymal stem cells are promoting cancer cell growth. We have EVs that are released from the cancer cells that can modulate the immune system. There's a lot of different examples across the general cancer hallmark pathways. And so for my focus on looking at resistance, there's the very straightforward mechanism of simply the fact that EVs are only the vehicle of transportation to be able to export drugs out of the cells. But my interest is more so on the cargo that EVs can carry that are activators of cell signaling pathways that are inducing phenotypic changes in the receiving cells. So these are going to be EVs that I'm focusing on that are released from the cancer cells that are then received by nearby cancer cells to either induce or maintain a drug-resistant phenotype. So in prostate cancer, we have a couple examples of this already. We know that EVs have been able to transfer resistance to docetaxel to other prostate cancer cell lines. This was potentially through the transfer of the multi-drug resistant 1 protein, but unfortunately there hasn't been a lot of follow-up to that mechanism yet. Additionally, in response to androgen deprivation therapy, we know that the protein cargo of LNCAPs, which is a prostate cancer cell line, has changed in response to this therapy and shifting. We see more glycolysis or other signaling pathways, which indicates that the EVs are changing and responding along with the cell of origin to these treatments. Additionally, inhibiting EV secretion sensitize prostate cancer cell lines to androgen deprivation therapy, and even EVs that were isolated from prostate cancer patients, these were able to actually induce expression of EMT markers in epithelial cells. So we have a lot of examples in which EVs are modulating the signaling events that are happening in the receiving cells and potentially progressing, having a lot of tumorigenic phenotype responses. And so the focus of my project is specifically on the proteins that are carried by these EVs that are coming from castration-resistant prostate cancer cells and impacting treatment resistance. So with EVs as a nanoparticle, the details of the isolation characterization can be incredibly important so that everybody sort of believes that I have the population I say that I have. All right, so just quickly over the basics, I isolate my EVs by differential ultracentrifugation, which basically means I take my cell culture supernatant and then go through increasing speeds to remove my unwanted large cell populations, such as cells, cell debris, apoptotic bodies, until I finally end up with my small extracellular vesicle population at the end. And so when I say small, I'm talking like 100 to 200 nanometers in diameter. I don't think this video is going to play for me. Oh, yes, there we go. And so we can use nanotracking analysis, which uses a laser, and then we have the light diffraction and the movement of the particles to estimate the size. And so this is just one example of my isolations from the cell line 22RV1s, where you see the majority of the EV population here, where we're looking at concentration of EV particles per mil, is between the 100 to 100 nanometers in size, which is what we expect. Additionally, we have expected protein markers for EVs. We expect enrichment in CD9 and TSG101, which are proteins involved in the structure and secretion of EVs. And so we have an enrichment for the prostate cancer cell lines that I'm looking at over from the cell lysates. Additionally, for a cell lysate marker, we've really found that EVs carry a lot of proteins from the cells of origin. So an actual just pure cell lysate marker is taken with a grain of salt, but it's still believed that calnexin, which is involved in protein folding in the ER, that this should not be found in EVs and has not been found in mine. So that was good news. Right. But I wanted to sort of broadly characterize in an unbiased way the EV proteome. And so I decided to do this using mass spectrometry. And so as a basic overview, I take my EV proteins, I then digest them down into peptides, and then clean them up to remove unwanted molecules such as DNA, lipids, or salts. I then run them through the mass spectrometry instrument and get these spectral patterns of intensity that we can use software then to pattern match the spectral patterns to that peptide ID, and then rebuild from those peptides the proteins that are found in my EV samples. So I did this with 9 different cancer cell lines in duplicate across the 3 subtypes of double negative, neuroendocrine, and then also AR positive. And I was impressed with the number of proteins that I was able to find for each cell line, representing that EVs have a lot of, a diverse array of proteins, and additionally, quite a few specific to the cell line and subtype, because only 2,000 of those were shared across all of the lines. And looking at we found 750 proteins that were significantly different between the subtypes even. So there's a lot of markers there for the EVs and potential. But keeping a sort of a broad overview look at the EV proteomes and what we found, we were also surprised to see how well they clustered by subtype. So this is a UMAP that showed a cluster of AR positive EVs, showing that they were similar to each other, except for LNCAP 95s, which is an interesting exception. Then we also have, the AR and neuroendocrine negative lines in their own little cluster. And then for the neuroendocrine positive, these two were incredibly similar as compared to the EF ones. So we find this particularly interesting because it represents that EVs are reflecting a lot of important distinctions about their cells of origin and keeping a lot of potentially of those cell signaling mechanisms that are happening in the cell lysates. And so we explored this further by asking whether they actually contained AR activity. So we took a traditional sort of RNA-seq type of analysis and took one of their signatures, since I had 6 of the 9 genes of that signature, and found that all of the EVs from the AR positive line scored really highly for AR activity. It was a little bit of a surprise how highly the AR independent negative lines, especially the double negatives, scored. And this seemed to be driven a lot by ACSL3 and FKBP5. Not sure what that means or why that is happening, so definitely something to explore. And it's also interesting that these 2 genes were not highly expressed in most of the AR positive lines. So we do need to refine this analysis. This is a signature developed from RNA-seq data, so it could be very different for the proteome, and then also EVs as compared to the cell lysates. But I think this is a very interesting question, and the fact that this can convey actual AR activity and cell signaling in the EVs. We did also look at other RNA signature cell lines, especially for luminal, basal, and neuroendocrine. So for a luminal signature, these are expected to overlap with AR positive and AR driven cancer. And so we do have 2 of the AR positive EVs scoring highly. From a basal perspective, we expect AR independent, and the double negatives did score highly, although oddly 22 RV1s, again, not quite sure what that means. So for neuroendocrine, one of the neuroendocrine lines, NCI H660s, that scored incredibly well. That's why it's shown in green. It kind of blew out the scale. So the other neuroendocrine lines didn't seem to score as highly. Again, these are signatures developed from RNA-seq data, and I only have a fraction of the genes that should be represented for an accurate signature. So we're looking again to refine this analysis for the proteome. All right, but then taking the next step in sort of focusing more so on now specific proteins that are enriched by subtype. So this is a volcano plot in which proteins that are more highly expressed in the neuroendocrine are here on the right, as compared to AR positive or double negative on the left. And so the one that I'm just going to briefly talk about at the moment is UCHL1. So UCHL1 is a deubiquitinating enzyme, and I thought it was particularly interesting as a neuroendocrine marker because it was shown to have a lot of involvement in neuronal specific cell signaling pathways. It's been briefly studied in prostate cancer and noted to promote metastasis as well as chemoresistance when overexpressed. However, when I looked more closely at my EV mass spec data, while this was expressed in all the neuroendocrine lines here on the left, we do also have expression in the D145 EVs, and this is recapitulated in the whole cell lysates where, yes, it's in the neuroendocrine lines, but also very strongly in the D145s. So it's definitely not a neuroendocrine specific marker, but could still be relevant to aggressive variant prostate cancer. And so what I want to do is move beyond characterization and look at the actual functionality of these proteins, and particularly UCHL1, that might be contributing to resistance. So unfortunately, I haven't had a lot of time to probe into this just yet. So right now, I sort of just have basics proof of principle and showing that my EVs are viable and capable of transferring proteins. So this Western blot here on the left is me adding GFP EVs to link cap cells and able to see 24 hours that that GFP protein was actually inside those cells. So at least for the EVs in my method of isolation, they're still viable, they can still transfer proteins. Additionally, I tried to recapitulate what was already known in the literature, that adding 22RV1 or DU145 EVs back to link caps increased proliferation. And so this is 6 days after adding the EVs, we did see an increase in proliferation. The expected hypothesis would be that 22RV1s are doing this through transferring proteins that are activating AR proliferation, and DU145s through some unknown AR independent mechanism. So there's clearly a lot left to explore about that, how EVs are actually doing this, and I'm looking forward to doing that in the future. All right, so that is all I have for you today, and talking about the summary or the proteome of the EVs and how they contain important information about the cell, including cell signaling events and also potential histology that can determine the cell of origin. Additionally, there are protein markers that are enriched in specific subtypes of EVs, although at least for UCHL1, it's potentially not as specific as what my analysis originally brought up it as. And then EVs are also capable of transferring proteins and inducing a phenotype that I'm looking forward to exploring more from the direction of UCHL1 and aggressive ovarian prostate cancer. I want to be able to identify more proteins of significance in the different subsets, and then also look at this from EVs being able to transfer resistance to androgen deprivation therapy. Okay. So thank you to Justin and the members of my lab for help on all these different avenues of my project, and also for making it a very fun research environment. So here we are at our last holiday party. Also, thank you to Justin Wong and his lab, specifically Abdur and Hannah, who helped out a lot with the bioinformatic approaches and the analysis of the mass spec data. Additionally, the nanomaterials lab and the CMSP had very important instrumentation that are necessary for this project. And thank you to the T32 for their years of funding. Okay, so with that, I am happy to answer any questions.
Thank you, that was really nice. So can you maybe explain a little bit about this, how the signaling pathways would be transferred? Do they have receptors in the membrane, or are they transferring the components of pathways, or is it known?
It's very difficult to look at exactly where the protein is in the EVs. People have tried, and it's very much dependent on the protein of interest. The expectation, though, is that if they are receptors, they can be on the outside as well, but could also be interior. Yeah, so both.
Are they loaded with phosphoproteins or activated kinases? Has anyone looked at that?
Yes, not a lot, because it is very difficult to look at phosphoproteins on the scale. It is something that I would like to do, but there is evidence that there are phosphorylated proteins that are actually carried by the EVs.
Other questions for Megan? Thanks.
So would there be a way to, like if you were to run this screen again, to add a selection pressure across your subtypes in order to enrich for, a protein or some other packaged element that would kind of differentiate between what you've already found at a baseline level?
Yeah, I mean, it would definitely be possible. Yeah, it could be very interesting. Part of it is that a lot of the cell lines that we have, they're not very responsive anyway to androgen deprivation therapy. So there wouldn't be enough selection pressure at least for a lot of the cell lines that I currently have from that angle. But if I had a panel of sensitive cell lines, yes, I agree that would be a very interesting experiment to run.
Or can you put the cells in like a scenario where like the NEPC cells thrive compared to like just a steady state cell culture condition, and then those proteins that are important for growth in that scenario would kind of enrich in the EVs.
Yeah, off the top of my head, I'm not sure what environment that would be necessary, but
Sure,
yeah, I
I'm
agree
just-
that that's a very interesting direction to go and could, yeah, the changes in EVs and the signals that they might be promoting would come across that way. I agree. Yes, thank you.
All right, let's thank her one more time for a great talk.
No, that ain't it. Sorry. Right? No.
I think it's hidden by. Yeah, right there.
Quit. Where is it? Okay. All right. Should be good. Nope. Still says we're not sharing. Where'd it go? Tell me this. All right. I don't know where it went, but it's somewhere. Should be there.
Hi, so I'm Julia Austin and I'm in Carol Lang's lab. And so I want to talk about my project, which is on the glucocorticoid receptor as well as the mineralocorticoid receptor in breast cancer. And so it's just a little bit of background on breast cancer, so breast cancer is characterized by a hormone receptor status. And so for luminal breast cancer, it's characterized by the presence of estrogen receptor and the progesterone receptor. And luminal breast cancer tends to have the better prognosis compared to triple negative breast cancer. And luminal breast cancer is characterized by two subtypes, luminal A and luminal B, with luminal B generally more proliferative than luminal A. And then with triple negative breast cancer, it's hormone receptor negative, so it does not contain the estrogen receptor, nor does it contain the progesterone receptor. And then these tumors tend to be more aggressive and have a poor prognosis compared to the luminal breast cancer group. And so for my project, we're primarily focused on the triple negative breast cancer subtype so far. And so some factors that contribute to triple negative breast cancer incidence and mortality is age, race, genetic predisposition, obesity, being pregnant for the first time after the age of 30, and then chronic life stress and social isolation. And so some mediators of the stress response are corticosteroids. And so these corticosteroids bind to two hormone receptors, the glucocorticoid receptor and the mineralocorticoid receptor. And so the main function of the glucocorticoid receptor, which I will refer to as GR throughout the rest of this presentation, is to regulate inflammation and metabolism. And then the main function of the mineralocorticoid receptor, which I will refer to as MR throughout this presentation, is it modulates ion transport and homeostasis, but MR also functions to modulate tissue response to injury, as well as early cell response to stress. And one of the things about MR and GR is that they also share some of the same ligands, which can kind of make it difficult to determine which receptor is being activated if you're looking at them at the same time in the same cell type. And so previous literature has shown that GR is elevated in triple-negative breast cancer. And so patients that do not have the estrogen receptor expressed and have high levels of the glucocorticoid receptor expressed have a lower chance of relapse-free survival compared to patients who express the estrogen receptor and then have higher levels of the glucocorticoid receptor. And in the study, it was shown and determined that up to 40% of triple-negative breast cancer express elevated GR. And it's also been determined that GR expression is associated with chemoresistance and metastatic recurrence of triple-negative breast cancer. On the other hand, MR is not widely studied in breast cancer. There have only been a couple of literature on MR and breast cancer and a couple in the triple-negative breast cancer subtype. And so what has been determined is that in triple-negative breast cancer, MR expression is associated with the triple-negative breast cancer phenotype. As we can see in that patients that are ER negative, it has higher expression of MR compared to patients that are ER positive and don't have high expression of MR. And another study showed that MR is mainly cytoplasmic in triple-negative breast cancer tumors. And so what we are interested in is determining what the role of both GR and MR is in triple-negative breast cancer. And so the first thing that we did was looked at data sets to see if the combined high expression of MR and GR had an effect. And so what we saw is that in ER negative patients in a GEO data set is that it seems that a combined high expression of MR and GR predicts a worse outcome compared to patients that have low MR and GR. And we've also looked at luminal breast cancer as well. And so what we found is that compared to the luminal A, it seems that combined high expression of MR and GR predicts worse overall and relapse-free survival in luminal B breast cancer. And so taking this information, we've come up with a few hypotheses to determine the effect of GR and MR on triple-negative breast cancer proliferation. And so what we have already known with information that's already come out of the Lang lab is that TGF-beta, which is a stress signal cytokine, it activates the P38 MAP kinase pathway, which then will phosphorylate GR. And then that will activate genes that cause the proliferation and carcinogenic potential of triple-negative breast cancer. And so we are interested on if MR does the same thing. And so we are interested in determining if TGF-beta treatment, if that activates the P38 MAP kinase pathway, and then that could potentially phosphorylate MR and cause the carcinogenic potential of triple-negative breast cancer. And then we're also interested in if there's an interaction between MR and GR upon TGF-beta signaling, as there have been previous studies in different cell types that have shown that MR and GR do interact and form a heterodimer. And so we are interested if that same thing occurs in triple-negative breast cancer, that can also lead to the carcinogenic potential of triple-negative breast cancer. And so this is a new project in the lab. And so one of the first things that we did was determine what cell lines that we needed to use. So we looked at both triple-negative breast cancer as well as luminal breast cancer, and we first looked at this cancer cell line encyclopedia and looked at the expression, the mRNA expression of both GR and MR. And so we've determined that the 231 cells have high expression of GR and also some expression of MR. So that's one of the models that we have decided to move throughout, to use throughout this project. And then for the luminal breast cancer types, we've decided to move forward with the BT474 cells. And so we also confirmed the mRNA expression from the cancer cell line encyclopedia via Western blot. And so what we saw and found is that MDA and B231 cells do express MR as well as high levels of GR. The T47D cells also express MR and GR, and then the BT474s express MR and GR. So these are just some of the models that we will be using for this project. And then we were interested in looking at seeing if corticosteroids as well as TGF-beta are able to stimulate migration in the 231 cells. And so we already knew that dexamethasone was able to stimulate migration in 231 cells, but we were unsure if aldosterone was able to do it. So dexamethasone is just is known to be a GR agonist, and aldosterone is known to be an MR agonist. But from our studies, we've also determined that they can actually both bind to both the MR and the GR. And so what we found is that aldosterone is able to stimulate migration in the 231 cells, and dexamethasone is also able to stimulate migration in the 231 cells, and then TGF-beta, which activates stress signaling, is also able to stimulate migration in the 231 cells. And so the next thing that we were interested in is determining if this migration in the 231 cells was through the mineralocorticoid receptor. And so the way that we determined to do that was by knocking down MR via transient transfection by siRNA. So on the left here is just a Western blot showing that MR is indeed knocked down after we transfected the siRNA into the 231 cells, and showing that GR is not affected. And so what we determined is that in cells that were not transfected with siRNA, dexamethasone and TGF-beta still stimulate migration in the 231 cells, and then in cells that were transfected with the siRNA, dexamethasone is able to stimulate migration in the 231 cells. Aldosterone does not stimulate migration in the 231 cells that have MR knockdown, and then neither does TGF-beta. So this is indicating that the TGF-beta migration in the 231 cells seems to be through the MR signaling pathway. And so another pathway that we are interested in is determining if MR and GR is able to interact in the 231 cells. And a way that we looked at that is through proximity ligand assay. And so we also treated the cells with both aldosterone, dexamethasone, and then aldosterone plus dexamethasone. And so what we see is that it seems to be that aldosterone and dexamethasone alone seems to cause MR and GR to interact. But when we combined aldosterone with dexamethasone, there seems to be more of an interaction in the 231 cells. And this interaction also seems to be more nuclear. And then we also wanted to see if we could have MR and GR interact upon TGF-beta treatment. And so what we see is that TGF-beta treatment alone causes the interaction of MR and GR, and which we found to be really interesting. And also too, it's possible that it seems to be more cytoplasmic and not as nuclear as the aldosterone plus dexamethasone treatment. And so that's what we have found so far, is that it seems that TGF-beta is causing migration in the 231 cells through the MR pathway, and is also causing MR and GR to interact. And so since this is a new project, we have more directions that we need to move in. And so we are we're wanting to look and see if MR is also involved in breast cancer progression in luminal breast cancer cells, and also wanting to further look at seeing if stress signals are activating MR, and then furthermore wanting to see if MR is also required for phospho-GR actions, and then hopefully eventually doing an RNA-seq and RPPA analysis. And so with that, I would just like to thank the Lang Lab as well as Cy and Caroline, who helped with a lot of the experiments, and then the 232 for funding, and I will take any questions.
All right, great. I'd like to start with a virtual question. I did just a message from Dr. Cui that he'd like you to contact him. Does anyone have a question here in the room? All right, hang on.
I was just curious about the two bands that you see in the 231 for GR. That was pretty prominent compared to the other cell lines. That was my first question, just what that second band is. And then have you looked in the HS578Ts at all? Because it looked like you had similar expressions of GR and MR in those as well.
Yeah, so with the two bands, so GR has multiple isoforms, and I think the top band is GR alpha, which some antibodies you can see both isoforms, GR alpha and GR beta, they're around the same size. So it's possible the second band is just GR beta. It also could just be a non-specific band as well. And then we have looked at the HS570s a little as well.
Does it have that second band also? I mean, it's kind of interesting that it's that one cell line versus
Yeah,
the
and
others.
so I've seen not all cell lines contain both of those GR bands. Some only contain like one, and then I've seen some contain the other ones. Like Ovcar, in my hands, I've seen with Ovcar 5 also, which is an ovarian cancer cell line, also contain both bands. But I think 231s also have a whole lot more GR than the other cell lines as well.
All right, if there aren't any more questions, let's say thank you one more time for a great talk. Thank you.